Yeast extract is a concentrated form of solubilized cell constituents resulting from autolysis of yeast cells with the cell wall removed. Yeast extract is rich in amino acids, carbohydrates, vitamins, and minerals. Therefore, it is widely used in food processing industry or as nutrients for bacterial culture media. Other than autolysis, extraction methods such as plasmolysis and hydrolysis are often used. The plasmolysis process uses chemicals such as salts to create osmotic pressures or ethyl acetates to alter cell permeability to enhance the extraction. Hydrolysates are prepared by treating slurry of inactive dry yeast with hydrochloric acids in different concentrations, followed by cooking, cooling, neutralizing with sodium hydroxides, filtrations, and concentration to paste or spray drying to powder. These two methods have limited use in the food industry because of the high level of salt presence and the destruction of certain amino acids and vitamins in the final product. In food processing, we tend to increase the concentrations of guanosines monophosphate 5' GMP and inosines monophosphate 5' IMP, which have the flavor-enhanced properties. We tend to minimize the concentrations of lower molecular weight non-flavorizing substances such as 5' nucleosides and 3' nucleotide. The diagram shows the overall process of yeast extraction. First of all, spent brewer's yeast slurry is depleted by removing the beer liquor. It is heated to between 40 degrees Celsius and 55 degrees Celsius, and the pH is adjusted to 6.2 to 6.4 for optimum autolysis. At that condition, yeast cell is killed and it promotes the activity of intracellular hydrolytic enzymes such as protease, carbohydrates, and nuclease. Cell death brings about disorderliness, resulting in the indiscriminate action of the hydrolytic enzyme on the cell constituent. The intracellular proteolytic enzyme degrades protein into peptides and amino acids, and the small amounts of ribonuclease acts on the RNA to produce ribonucleotides and ribonucleosides. The high organized cell membrane begins to degrade and lose its integrity, thus releasing the soluble into the aqueous environment. Autolysis may sometimes be accelerated by the addition of exogenous enzymes such as papain and glucanase. These exogenous enzymes attack directly on the yeast cell wall, causing the cell wall to become porous. The cytoplasmic content of the cell, including endogenous enzyme, would be leached out to permit better interaction between the substrates and the enzyme. Besides that, the enzymes are small enough to penetrate through the yeast cell walls and act hydrolytically on the intracellular component. Protease breaks down protein to form smaller constituent units such as peptide and amino acids. The autolyzed yeast contain 50 to 80% of intracellular RNA. 5' phosphodiesterase is added to hydrolyze the extracted RNA to 5' nucleotides. Adenylate deaminase from Aspergillus oryzae is added to hydrolyze 5' AMP to 5' IMP. 5' phosphodiesterase is obtained from the extractions of malt rootlets. However, 5' nucleotidase, which convert 5' nucleotides to 5' nucleosides, has also been found in malt rootlets. It is inactivated by the heat treatment at temperature of 65 degrees Celsius for 30 minutes. After the yeast cell is autolyzed and hydrolyzed, it is sent for purification. The autolyzed yeast is purified to separate yeast extract from the remaining cell mass. Biomass separation is done by multi-stage nozzle separation to remove the spent cell fragment and other suspended fines from the autolyzed yeast. After that, clear extract is evaporated to paste or spray dried to powder. The yeast extract production by autolysis is considered safe as it does not produce any harmful chemical or pathogen. The use of yeast cells of Saccharomyces cerevisiae is generally recognized as safe. According to a conventional method, 5' phosphodiesterase is extracted from Penicillium citrinums or Streptomyces aureus. This method is not economical as the enzymes employed are extremely expensive and do not have green and safe status. Therefore, malt rootlets are used as a safer and more economical alternative. Yeast extract is produced commercially by autolysis. However, it has some disadvantages, which is low yield, difficulty in solid-liquid separation due to high content of residue in the autolysates, poor taste characteristic as a flavor enhancer, and risk of deterioration due to microbial contamination.
Created using Powtoon.